today I found out that I made a big mistake in my life and it's not coming earlier. I would like to thank especially the students for, especially the students for inviting me, and I think it's a wonderful opportunity to be able to organize these meetings for students. And it's also, of course, a wonderful opportunity for us to share the data with you. What I found the last sort of half day is a tremendous curiosity of your field. And of course, curiosity is a major drive for fundamental research and research in general. So, that's by far the most important drive. Sometimes in your life, you stumble upon a potential application which might have a big societal impact. And then I think also as a scientist, you need to grab that opportunity and be able to at least try and make that societal impact. And this is what happened when I stumbled basically upon the culture of project. And I must apologize, there's not a whole lot of molecular biology in my talk, and there's no genetics in my talk. So it's basically my task to sort of give you a window to applications of the technologies that you are learning and you are experiencing. So why would you want to create alternatives for beef? Not many people will realize that we will soon are facing a meat crisis. The demand for meat is going to double in the coming 50 years or so. Oh, the animation doesn't. So what is happening? The meat comes from cows and from pigs primarily, also from chickens. But let's keep to these examples. And these are very inefficient animals. Every 15 grams of edible meat, you have to put in these animals 100 grams of vegetable proteins to get that. So it's a bioconversion rate of about 15%. And in 2050, the consumption is going to double. And we are already using 70% of all our arable land, of all our agricultural land to produce meat. And actually, livestock meat production is going down instead of coming up. So we will eventually need an alternative to meat production. In addition, it's now known that cows and pigs and also chicken are responsible for about 20% of all our greenhouse gas emissions. So it's a major contributor to pollution and greenhouses. And it's primarily methane, a little bit less CO2, and also nitrous oxide. And of course, there are animal welfare issues, and whether you think that's important or not doesn't matter, but there are a lot of people who think it's important. And so that would also be a reason to start and think about alternative ways to do things. Now, how does the technology that you are learning and that we have available right now and that may basically come from sort of medical tissue engineering experiences and research, how does that help us in producing meat? I'm doing something wrong here. Okay. Thank you. Well, it's not a new idea. Already in 1932, Winston Churchill came up with the idea of, or at least with the concept of growing meat outside of the body. And he asked the question, why do we grow an entire chicken if we only eat the breast and the legs? It seems like a waste of energy and resources. Of course, he was a statesman. He was not a biologist, but he was befriended to a Nobel Prize-winning physiologist, Alexis Carrel, who at that time, for the first time, could keep tissues alive for about a day or 2 days outside of the body. And that triggered his imagination and said, well, if we can do that, then we may be able to create new tissues outside of the body in vitro and do something for instance, food production. So that was way ahead of its time because there was no technology yet to produce those tissues outside of the body. Nowadays there are, and again, as a result of tissue engineering, mostly for biomedical purposes. And so what we are proposing is to take stem cells from muscle in cows, and you can do that through a biopsy, like shown here, where you get a couple of hundred stem cells out of those tissues. And these are stem cells, it has already been alluded to, they're tissue-resident stem cells, satellite cells. Every skeletal muscle fiber has them. They're sitting there waiting for an injury to occur so that they can repair that injury. And you can take out these stem cells and grow them. Of course, the replication is an important part of it. So you take out this little piece of muscle, and that, of course, if you put that under the microscope, you will have both muscle cells and fat cells. The order is not, the ratio is not completely fair here. We also have fat cells. I will come back to that later. Also contains stem cells, but it contains, the muscle fibers contain these satellite cells that are PAX7 positive, and I'm sure after the break you will get much more insight into it than I can provide. But these cells, if you extract them correctly out of that muscle tissue, they will grow out and they will be able to multiply. And of course, multiplication is an important aspect and an important requirement for the creation of beef, because you need a lot of cells. So from one stem cell, we can theoretically get to 10 to the 14th cells, and if you do the math, that ends up being about 10,000 kilos of meat from one single stem cell. It's a little bit theory, but we are getting there in terms of these type of numbers of doublings that are required to get there. So muscle cells, as you probably know, are mergers of larger, of a lot of cells. They're syncytia, basically. So the first step of differentiation, and again that will be described more in detail after the break, I guess. The first step in the differentiation is that they need to merge and form so-called myotubes. And these myotubes, you can easily see them under the microscope. They have multiple nuclei. And the other interesting feature of these skeletal muscle cells is that they are capable, like most mesenchymal cells, by the way, are capable of self-organizing a particular tissue and forming in vitro a muscle strip if you provide anchor points. And here in this example, you basically see Velcro, which we buy from the local store. And if you put them in a gel in between those Velcro points, they will start to organize that gel and they will start to form a muscle fiber, all by themselves. We don't do anything about it. We just basically starve them and then they start doing it. So those features of those stem cells make them potentially an ideal source to create muscle tissue that eventually can turn into beef. So they bulk up because in between those anchor points they start to develop tension, they start to perform labor, which is of course important for their protein synthesis, and when they do that, they do indeed increase their protein synthesis and they bulk up and provide a pretty good protein source for meat. So that will turn into muscle tissue. And if you just take, this is the very simple approach that we have taken. We have grown literally for 120,000 of those strips, put them together in a hamburger, and cook it and eat it. And the purpose of this particular, I will come back to it later, but the purpose of this particular strategy is somewhat strange because we found it, because the idea seems crazy and sort of something sort of too futuristic. So we found it very hard to get funding for this type of research. And so we decided at some point to take a different approach and basically go out there and say, well, you know, we're going to make a proof of principle, and we will show to the world that it can be done. And that may change the whole discussion on, you know, from a crazy idea from somebody in a very small country that you don't hardly know that it exists, to it is a product, it's getting there, and we need to think about how to use this and how to improve it. And of course, there are meat substitutes from vegetable proteins galore. We all know them. Who of you is vegetarian? Wow, only you are in a minority. So that's typically in developed countries about 3 to 5% of the population is vegetarian. I guess here it's a little bit less. So it's, in my mind, it's not realistic to assume that this is going to be a major solution to the meat problem, that we all become vegetarians. However, there are a number of substitutes, meat substitutes, that are based on vegetarian proteins. They're pretty good. You know, they are edible, and we eat them quite often, but they are not meat. And we are working under the assumption that people want to eat meat, especially apparently meat turkey. So we need to make a product, a skeletal muscle that looks and feels and tastes exactly the same as the original product. Now that assumption can be contested, and it has been contested many times, as I can tell you. But it's still, I think it's still holding, and what's more, the World Health Organization thinks that by 2050, the meat consumption is doubling. So, but in order to get there, we need to improve the efficiency, and all of you who have done cell culture know that it's not a particularly efficient process, and it also provides sort of tissues that don't seem immediately appetizing. So we need to have two things. We need to make an efficient process, and we need to have mimicry. I apologize, I couldn't find a substitute for Prime Minister Erdogan. But we need to create something that is very, very similar, if not identical, to the real meat. Okay, so how are we going to do that? We have everything in cell culture, and the beauty of cell culture is that you have a lot of variables under control. You can play with the medium, you can play with the direct environment, you can play with conditioning, with temperature, with all sorts of variables to figure out what the most efficient way is of growing these tissues. So medium is going to be an important aspect, and currently we are using very traditional cell culture methods to culture these cells, so we're still using serum. And of course, we want to get rid of the serum, because eventually there will not be enough, if this is successful, there will not be enough serum around, because we are reducing the livestock, and the serum comes from the livestock. We will not have enough serum to produce these tissues, so we need to get rid of the serum. Also, these mesenchymal cells, as skeletal muscles, grow on surfaces, right? They don't grow in a solution, they grow on surfaces. So that's a limitation, because we all think in 2D cell culture, but eventually that's going to, and I will show that, it's going to require an enormous amount of surface to grow all these cells. And if we can change that stage early on from 2D to 3D, we will gain a tremendous efficiency and tremendous space reduction. So that's what we are working on. Also in terms of the medium, we have combined saltwater algae, and we have saltwater galore on this planet. Saltwater algae, and get the proteins and the carbohydrates from those algae, and turn them into an algae soup and feed them to the skeletal muscle cells. And if you combine those two processes, you potentially can gain tremendous efficiency and actually take CO2 out of the environment. And of course, what's what's not being done in cell culture at all right now is that you can recycle and regenerate the media that you are using, because the cells are not using every component of that medium, right, or not. So, of course, you need to feed the cells, you need to feed them sugars, proteins, and potentially fatty acids, which also might give the opportunity, because you have those variables under control, that you can create also omega-6, omega-3 enriched fat tissues so that a potential product becomes healthier for you. But for now, we need to just improve the efficiency. So this is what is required for our hamburger, about 60 billion cells, which is a lot of cells, I can tell you. And we are now using these cell factories, which are 10-layer cell factories, which have a surface of only 0.6 square meters. And we need about 200 of those bubbles, and that's all plastic, right? About 200 of these bubbles to create one hamburger. That's about it. We are currently, because we are doing this large-scale production and we are just an academic lab, so we are running out of storage space to get all the supplies in. So we need about this size of a room to get all the plastic in storage for this little hamburger. So just to illustrate to you that it's not an efficient process yet, and it's not a real product yet. Eventually, we're going to move to large bioreactors, and the largest bioreactors right now that exist for cell culture have a content of about 25,000 liters, which is a sizable swimming pool of medium. But that we have done some calculations of that. A bioreactor like that could feed 40,000 people eventually. So if you move from this type of system to this type of system, you already gain quite a bit of efficiency if you can completely bypass the 2D cell culture and start with 3D immediately, while retaining the proliferative capacity. You will gain another big, you will have another big efficiency step. So this is what it looks like. We are a small country, so we have a short supply of these incubators. We have these large incubators that are 4 times the size of the regular one, and we bought all these incubators in the Netherlands of this size to do this project. And these are all these 10-layer bubbles to produce eventually those 60 billion cells. This is a regular size incubator. And now all the tissues are in a 3D setup. So you have already from 2D to 3D a gain in efficiency. So this is just to illustrate how you could use the salt or freshwater algae to feed the cells. We are doing some work on that, but it's still preliminary. You have all those variables under control in cell culture, and one of the downside is that there are a lot of variables, so it's a lot of work and a lot of resources that go into this. One of the aspects where we looked at was changing the elasticity of the substrate on which these cells are growing, and the elasticity, if you have followed bioengineering classes, the elasticity of a skeletal muscle cell is about 20 kilopascal. So you would think that that would be the most efficient surface on which they grow, which would be a big pain because creating those elastic substrates is kind of cumbersome. So we looked at proliferation rates as a function of elasticity, and fortunately, on the infinite elasticity of a plastic, they grow fast. So that was helpful to us. And they do have a minor improvement at 21 kilopascal, and below 3 they basically don't do anything. So we were pretty confident that that we could continue doing this on relatively stiff surfaces. In addition to efficiency, you also need to mimic the tissue in terms of taste, color, and texture. And that's going to be a major challenge as well. So we have done some work on it. This is a cow. It's in bad shape. It has not trained very well, and it was not fed very well, and we need to go from there to here. This, by the way, is a specific strain of animals in Belgium, just south from us, that has a mutation in myostatin. And myostatin is the natural protein that limits hypertrophy of cells. So we are taking stem cells from these guys to see whether we get even more protein per cell out of the tissue. As I mentioned, these cells have the capacity to self-organize tissue. So if you put them in a gel in a petri dish, now the anchor points are not Velcro, but they are silk wires. Within 24 hours, it looks like this. The cells have self-organized into a muscle strip in between those anchor points and start to produce more fiber and more protein because they create tension in between those anchor points, and at some point they also pull themselves apart from those anchor points, so there is a limitation on what you can do. But they perform labor. It's static labor, but they perform labor, and it improves their protein synthesis. And we thought in our naivety that if we would stretch them even more and cyclic stretch as we are, and the cows are continuously moving, that we could improve that efficiency or that protein efficiency. And to our surprise, and this is at least some molecular biology for you, to our surprise we found quite the opposite. If you stretch these cells, these are cell lines and these are the primary cells, these are from mice, but it doesn't matter. You see that in the more mature myosins, actually expression goes down if you stretch them, which seems a little bit counterintuitive, but I talked to a number of neurologists who are specialized in neurodegenerative diseases, and completely denervated muscle, if you stretch completely denervated muscle in people, it also gets worse. So in real life, we never have the passive stretching that you sort of simulate in a laboratory with a flexor cell apparatus. So for these particular cells, that doesn't really work. The other thing that you can do is electrically stimulate them, basically zap them with an electrical field. And that works. They start to contract. And they start to look like real skeletal muscle with their typical cross striations, which is an indication that they really have a high level organization of actin and myosin protein production. But of course, you can already figure out that this requires a lot of energy, and is probably not going to increase the efficiency of the whole program. And what's more, if you look at this contracting muscle strip. What's more, if you look at the effect of the electrical stimulation, it's there, but it's not a major leap forward, and you have to apply a lot of energy. So this is probably not the way to go. And if you think about it, it also makes sense because our muscles are not electrically stimulated in our body; they are chemically stimulated. So one of the steps that we will do in the next phase is try to give them little pulses of acetylcholine and see whether we can recreate that effect at a much higher efficiency. If you do that, so it is more efficient if you look at the more mature myosin heavy chains with electrical stimulation, especially if you start early, it increases quite a bit, and it also increases protein synthesis. So it does work, but at the cost of efficiency. Of course, there are other proteins in skeletal muscle that may be important or are important for color and may be important for taste, and one of them is myoglobin. And myoglobin, as you know, is sort of a correlate of hemoglobin, and in high oxygen environment, it turns red or pink. That's why our meat is red or pink. It's not because it contains so much blood, because it doesn't, but it's because of the myoglobin. And this also is an iron capturing molecule, very similar to, or the same as hemoglobin, because it has a heme group. And so it could add a lot of nutrients, color, and bivalent iron, which is the best iron in our diet to become absorbed. And it may also, that's my feeling, it may also affect the taste. So can you play with the myoglobin expression? And yes, you can, and actually in very, very simple ways. If you culture them for 24 hours under normoxic conditions, so at 2% oxygen or at 1% oxygen, they will start to increase their myoglobin expression about 5-fold. In the literature, it was described that you could also use caffeine, which is kind of interesting because you would eat hamburgers that would keep you awake, but some people then would not be able to eat them in the evening, so only at breakfast. But in our hands, it didn't work very well, so we forgot about that. But the oxygen manipulation basically changes myoglobin levels, and these are very, very simple interventions in a cell culture, even in a large-scale cell culture. So right now we are here. This is our cultured beef. This is bovine muscle cells. This is about 1,000 of those muscle strips. You can see them pretty much individually. And this is our mini burger. This is our pre-beta version. And we cooked it and ate it, and it looks like this. And it actually tastes like a hamburger. So I thought that's not very difficult to get it to taste like a hamburger, but it does. So we are pretty confident that we can come up with a prototype with a proof of concept hamburger to show to the world as a example of that it's possible, as an illustration that it's possible. Now you would argue, this is not only skeletal muscle. In fact, meat is not skeletal muscle; it's dead skeletal muscle, which is another issue. But there will also be fat in there that will be important for taste and important for texture. And there may be bones in there, and maybe you want to eat a shank, and then it has bone marrow. Can you create that? Of course, yes, you can. From mesenchymal stem cell sources, you can create all these tissues. They're all being described. In fact, it's sort of a requirement in research to identify a cell as a mesenchymal stem cell that you are capable of driving it into the adipogenic pathway. So you can make this adipose tissue. And in fact, we have done that also, and other people have done that too. This is not from our laboratory, but from somebody else. You can make those fat tissues from adipose tissue-derived stem cells from the same biopsy that you took the skeletal muscle from. However, the problem currently is that the described methods to create those fat cells require high dosages, high concentrations of dexamethasone, which is a steroid, which I don't want to have in my food, and IBMX, which is a super caffeine, which is actually toxic for people and has not been developed afterwards other than a research drug. So, fortunately, the biochemical pathways through which these triggers work have all been described, and there are common pathways that you can use in the PPAR area and the CPD area where a lot of research has to go into it, and we will probably at some point be able to bypass the initial dexamethasone and IBMX triggers to create those fat cells, but we're not there yet. Then, as you probably know, we are limited to small samples because the tissue, it's living tissue, it's metabolically active tissue, it's also mechanically active tissue, so it requires constant nutrients and oxygen delivery. And of course, it's all dependent on diffusion, so there is a rate limiting step there, and at some point, with a given thickness of the tissue, the inner part will die because it will not have enough oxygen and nutrients. So that's why we have blood vessels, as earlier explained in tumors, and that is sort of standing between us and making large, thick cuts of meat, like a steak, not a hamburger, but like a steak. Mind you, processed meat is 50% of the meat market, so if we have solved that problem, we already have made a big leap forward. But of course, my ambition goes a little bit further. I want to make a complete steak. And then you would need to create either blood vessels, which is basically my primary job. This is just a hobby. Or you would need to make a channel system through which the nutrients and the oxygen can flow, which is probably more realistic in doing that at this point in time. And right now, as you probably have heard or have seen, is that there are lots of 3D printing methods out there that can print matrix in every type of shape or form or configuration and cells at the same time. So eventually, I'm pretty positive, and we're talking to the bioprinting people about this, that we can combine these two technologies. It's going to be difficult, but we can combine these two technologies to make really thick cuts of meat. So then, of course, there are many, many challenges. I've talked about taste and not much about texture, creating large chunks of meat, more protein per fiber. We need to scale it up, and we'll come back to that later. And people need to start eating, right? That's the prerequisite of the success of cultured meat. And if you go through the streets, of course we haven't gone through Istanbul, but we've gone through London and Brussels and other cities. If you just go through the streets and ask people who shop there, are you interested in cultured meat, and do you see that you are going to eat this in the next 10 years or so, they invariably answer, are you kidding me? No way. However, if you give explanation on why we are doing this, and if you rephrase the question, if you say, well, let's assume 20, 30 years from now, you enter the local supermarket and you have these two products. They are identical. This one is made in the lab, hence this little label. The other one is made through livestock, which is now 4 times as expensive as we are experiencing right now because of the scarcity and because of an eco-tax. And it also has this nasty label on it, like familiar with, in cigarette packages, right? That the stuff kills you. This has killed other creatures on the planet. And if they're the same prices, or this is even cheaper, and they're the same flavor and the same texture, why are you going to do it? And I think eventually that can be made a pretty convincing story also for the general public. You have to find out, but I'm pretty confident that it will help. So that they will turn to large-scale eating of cultured meat. A life cycle analysis has been done. Is this really a solution to the environmental issues? And Hamakao Tuomisto has done some calculations with a lot of assumptions, admittedly. The land usage, of course, is going to go down tremendously. Water usage is going to go down. We need about 15,000 liters of water for 1 kilogram of beef. So that's coming down tremendously, and the energy consumption is going down tremendously. And one of the reasons why we have chosen beef, because these effects are largest for beef. They are less big for pork and even less big for chicken, for poultry, because poultry is much more efficient in bioconversion. That first hamburger we're going to present in the coming weeks, months to the press is going to cost, it's a quarter-pound hamburger, and it has cost 250,000 euro, which is roughly about 600,000 euro. So it's expensive, right? It's not really something that you would buy tomorrow and savor. And this is, of course, completely prohibited. But that's because it's man-made, it's made in an academic laboratory, and in a non-efficient manner. And I usually compare this with the first ENIAC computer that was built in the United States, or the Colossus in Britain, which cost about $2 million, had about half the size of this room, occupied, and 50 ladies typing in data every day. And it has less calculated power than your current cell phone. So those prices have gone up tremendously, and the performance has gone up. And recently we did an analysis based on processes. This is not really my work. This is work from somebody who is head of a R&D in a company that produces by far the most stem cells, cell adherent culture, or surface adherent cultured stem cells for medical therapy. They are basically doing all the cell culturing for the major companies that do clinical trials on stem cells. And they have a process described from starting with a single flask to a 1 liter bioreactor to produce the volume to feed the large 25,000 liter bioreactors. And he has a model calculating based on the numbers that we provided with our replicative capacity and our highest cell density per milliliter capacity. It's quite amazing that if you go through that process and do all the calculations, we come up with, again, with much more detailed information. That of course, the materials are by far the biggest portion of the cost. The facility is very little, quality control is very little, and labor is very little. And surprisingly, they came up with a cost per kilogram of beef production of about $65, which suddenly becomes sort of realistic. It's still very expensive, but this is without any improvement in the efficiency. This is only the cost factor. You still have that truckload of plastic. But it suddenly becomes a realistic number, and I'm sure we can get that down tremendously. So, this may be a possibility. This may be something that is going to work, and that creates an alternative to huge problems that we are facing in the coming 20, 30 years with livestock meat production. And I'm sincerely hoping that more people start working on this and that more resources become available, because there's still a lot of work to do. There's no question about it. But you have to start somewhere, and at least we have shown for now that in theory it's all possible. So these are the people who have done most of the work. I'm usually only talking about it. And it's in 4 universities, all in the Netherlands, my own universities, Eindhoven, Utrecht, and Amsterdam. We have a little foundation. This actually, Willem van Helen, is a 19-year-old entrepreneur. He's not even a scientist. He bailed out of medical school. But he was in a prisoner of war camp during organized by the Japanese, and suffered hunger. But also suffered a lot of animal cruelty, and he is very passionate about getting this going. So he actually, as a 90-year-old entrepreneur, convinced the Dutch government to put money in this, which is quite amazing. And he pulled together all these people to make this happen. Right now we are sponsored by philanthropists. One of the side effects of a subject that, although it's futuristic and kind of scary, it still gets a lot of media attention. And the media attention actually helped in getting philanthropic funding for this program. So we're very grateful that we can do this. We can produce this proof of concept, and from there we'll see how the reception is going to be and how we can continue this research. With that, I would like to thank you for your attention. I'm happy to entertain questions. If you want to ask them in Turkish, I will put on my translator, otherwise, it's wrong. Otherwise, I will do that.
Yeah, two points. You mentioned the use of, control of oxygen in your calcium. That is part of interest, because it wasn't clear to me what you consider as hypoxic or normoxic, because we don't really know the amount of oxygen that is in vivo in a muscle, and that is also of great interest.
Because when
I don't know one tissue. except maybe the aorta, or that we'll see 20% oxygen, that is the one we have in the atmosphere. Therefore, much lower concentration must be in action before. And we know that, for instance, in our hands, culturing the muscle cells in 3% oxygen helps a lot their survival and proliferation. So that might 3%,
right.
3%. Okay, that is one question. The other one I will be quick. I mean, you're focusing on the beef, but obviously there is chicken, pork, and lamb, which must be considered to in this type of approach. That I didn't know that you were doing such a thing. And I'm kind of surprised and, well, interesting.
Right. Well, yeah, so the first question, as I mentioned, we are using traditional cell culture methods, and it's correct. The oxygen levels that we are typically using for cell culture are most likely not physiologic. So some people try to reduce that systematically to 14%, for instance, which seems, it's kind of arbitrary, but it seems a bit more physiologic. At 3%, we haven't tried 3%. At 2%, yes, they do survive very well, but they also differentiate much quicker. So we need to, since we need to go through an obligatory proliferation phase, we need to play with that variable at what time we are going to produce it, because it does, in our hands at least, drive differentiation rapidly and steers away from proliferation. So that's why and in terms of other animals, of course, every animal, as far as I know, at least every vertebrate, as far as I know, has satellite cells in their skeletal muscles. We have tried mouse, pig, and bovine. And the processes are slightly different, but they are mostly very similar. So yes, you can translate it to others. And in Scandinavia, they're now doing fish. And one of the good targets would be tuna, because that's soon going to be extinct if we keep on fishing it. So there are lots of other reasons why, other than sort of food scarcity, why we would try and translate this to other species. Do I need to? Oh, okay.
First of all, I want to say welcome to Istanbul. Thank you so much. And I want to ask that what's the negative side of this meat? You don't speak about. Can it be like GMO? Negative? Can it make cancer? I want to learn this.
You mean this type of technology? Whether? I try to avoid the GMO issue as much as I can, because of course it's difficult enough to get this across. So, but there are many solutions to the food scarcity, right? Not as much for beef, but for food scarcity in general. I mean, we are throwing away 40% of all the food that we produce. If we start to change that, we can have also a major impact on food scarcity. So there are many different ways. This is just one of them. In terms of therapeutics, there's only one relationship, and it's pretty weak, between eating red meat and colon cancer. It's an epidemiological relationship. There's no, as far as I know, no molecular biology behind it. So I wouldn't know where to start to create a meat product that could avoid that sort of risk. But potentially, if you find that biology relating eating red meat with colon cancer, yes, then we could potentially do that. That would go into almost sort of a crossing between pharmaceuticals and nutrition, which of course is happening in many places and has been happening. I mean, we have added vitamin D to all sorts of substances, and we have added iodium to salt. So there are already nutraceuticals in our system for a long time. And we can think of extending it, but we need a lot more biology to understand the relationship between colon cancer and human growth. Do I need this? I do. I do.
Ben de teşekkür ederim sunduğunuz için. Bu üretilen kırmızı et bizim şu an yediklerimiz kadar besleyici olacak mı? Ya da yani bilebilir miyiz besleyicilerini?
Right. It never works if you answer it. We don't know yet. Eventually. Oh, sorry. Right. You should have a translator as well. The question. I will repeat the question. No, it's a good point. The question was what the nutritional value of this particular product is, and whether it's going to be the same as livestock produced meat. And eventually, it's going to be the same because these fibers, in the end, if we treat them correctly, are indistinguishable from meat fibers in terms of their protein composition, DNA composition, fat composition, minerals, etc. We are not there yet, but I'm pretty confident that we will get there.
Actually, I wondered, we are doing that, but how can I say? When we do...
You want to do it in Turkish?
We are doing that, but we will try to more protein or something like that. Also, when face the industrialization stage, they will try to add in something nutrients and chemicals. What we will do about that?
Right. This is actually a very interesting question because it expresses the fear that we all have, that if we move a food production from a quote-unquote natural production system to a factory production system, you will be vulnerable to nullified practices, vulnerable to mistakes that are being made, human-made mistakes with the food production. And I guess that's the general fear that people, not always as well expressed as you do, but will have. And I can only say that's a matter of, the only thing we can do about it is regulation. This is kind of an interesting spin to it. This was actually thought up by students. We do design projects with students from a design school, and they came up with an interesting idea. We have many problems with food. One is that current generations, and I'm not trying to accuse you, but it's just a general description, not always know where the food is coming from. And so you could combine those two with, assume that you have a community, let's say a neighborhood in Istanbul, and together you have a little farm with one cow, one pig, and one chicken, and you take care of that animal together. It's about 40,000 people in that community. You take care of that animal together, and every half year or so you put a needle in it, extract the stem cells, and you produce in the local factory, very controlled in your own sort of environment, produce that meat. Then you would gain control over the whole process and it becomes much more transparent. That could be, it's very futuristic, I agree. The other thing is, I don't tell that story anymore, but potentially you could do this at home, right? Stem cells survive freeze drying. So you could order stem cells through the internet, reconstitute them, and have a bioreactor at home, and create your own meat. Of course, you have to know 8 weeks in advance what you're going to eat. But that will also increase sort of the confidence in the program. But yeah, you're right. There's no way around it. Once you industrialize it, you're prone to or vulnerable to nullified practices and mistakes. Should I? Please. Do I need it?
Teşekkür ederim. Konumuz çok güzeldi. Benim sormak istediğim, şimdi bugün tükettiğimiz tavuklarda, broiler tavuklar çiftliklerde yetişiyor, güneş görmeden 45 günde kesime geliyor ve et olgunlaşmamış, lezzetsiz oluyor. Ama aynı tavuğu yine 45 gün içinde kendi bahçemizde yetiştirdiğimizde, güneş gördüğünde, hareket ettiğinde et lezzetlenmiş ve olgunlaşmış oluyor. Bu etin olgunluğunda siz kullandığınız elektrotlar kullanmışsınız, kasları hareket ettirmek için oksijen miktarını geliştirmişsiniz. Güneş ışığının da etkisi olabileceğini düşünüyor musunuz acaba?
That's interesting. That's a new question that I've never heard before, which is quite accomplishment. There's not a lot of information. There's some research on photon influence on cell culture. It's pretty obscure. There's no real hard evidence that photon exposure on cell culture has a real influence or has a predictable and reproducible influence. But there is some evidence. We haven't done any research on it. Of course, it's a slightly different answer than you require for your question, because you have a whole animal that's exposed to sunlight, right? And I'm not sure. I don't know. I'm not a veterinarian. I'm just a regular medical doctor, and I don't know what the influence on sunlight is on chickens. I assume it's very little because they have these feathers and very little exposure to. And as far as I know, they cannot get sunburn. But it may affect the produce that they're eating, and that way it could have a lot of effect on the meat quality. There are probably other variables as well, what kind of room they have to move and what they're being fed. And of course, in vitro, we have a lot of more variables that we can play with to eventually affect the taste and the texture of chicken as well.
Firstly, I want to thank you for your presentation. I want to ask you about, will it be accepted by the tissue using non-saturated fatty acids in vitro, such as omega-6 or 3, as you mentioned? Will it be accepted?
Would it be accepted? I think so. One of the most successful butter products in the Netherlands is a butter product that has been enhanced with omega-3 fatty acids. I think that has a big branding name and a big branding sort of benefit. So I think it will be accepted. Of course, if it starts to taste like fish, then it's a whole different matter. But if it doesn't really affect the taste, and in fact, it's likely not going to affect the taste if we have pure grass-feeding animals, like in some of the pandas in Uruguay. Their fatty acid content is much higher in omega-3 fatty acids than if you have feed animals in North America. So yes, the biochemistry of the cell, even in cow, is capable of changing the fatty acid metabolism towards more polyunsaturated fatty acids. There was a question about that. This is a question about that.
Okay.
Do I need it?
If you don't want to listen music, no, you
There's no music in it.
Thank you for the speech, first of all. I'm concerning about that situation. You mentioned that it is 4 times cheaper than produce this kind of beef. But what if, when you succeed, the scientists or merchants has said, yes, gentlemen, we produced it in the labs and it's 5 times more expensive than the normal bead when you succeed then.
Right.
What if it is being said that it's more expensive?
Right. I cannot really predict the cost. I had no intention to do so. The only thing that I wanted to do is to show you that eventually it's possible to come down with price into a area where it suddenly will become acceptable, at least to some people. And these calculations have been done under very very conservative conditions. So, for instance, the carrier beads, we had a price for the carrier beads that could come down easily 50% if you do large scale. So this is not going to be the definite price. I think it will be cheaper and cheaper. But eventually, I don't know where it's going to end. What we do know is that the price of regular livestock produced beef is going to increase. There's no question about it. It's way too cheap right now. We don't experience the meat prices because the meat price is artificially low, and that has to go up in the coming years, if not the next half year. I don't know who was first, but you were first.
You say stem cells tend to grow as monolayer as growing, but you also say we have to convert them three-dimensional structure. So my question is how this process works, or how you place the blood vessels or channels in the meat.
Right. We don't know yet, but there are, in tissue engineering, several techniques have been described to do that. You can grow the blood vessels. First of all, blood vessels are a highly specialized structure to transport blood. And blood is a highly specialized material to transport. It has the nasty sort of characteristic that it clots, which is fine because if we have a bleeding, we at least not bleed to death. But from a tissue engineering perspective, it's kind of nasty. We don't have blood here, so we don't need those very specialized structures. We just need channels. So I'm proposing that we use hollow tubes in a branching pattern made out of polymers, sugars, what have you, and then print them and print the cells in between.
Are they selected for nutrients?
In terms of the permeability?
Yes.
No, they're not going to be selected. But in cell culture, we don't have that selection criteria anyway, because we just now pour the nutrients on top of the cell. You might make them selective, but I don't think it's necessary in the beginning.
Thank you.
Thank you. I just want to ask that what is the specialty of blood vessels in our body that you want to create the artificial blood system? If you just do it with the old traditional way, pipetting or just giving the oxygen in a gaseous form, why it doesn't work? And do you really think that it really doesn't work and you need the blood vessels, artificial blood vessels?
I really think it doesn't work because we have tried. I don't know whether it has been explained before, but in our body, every cell is, the farthest it is away from a blood vessel is about 150 microns. Mostly, the largest is 150, 200 microns. Most cells are much closer to that. A cardiac muscle cell, which is highly metabolically active, is surrounded by 6 capillaries. The distance is basically zero. So it depends on the metabolism of the cell, but these cells, we know by experience that if you make the tissue thicker than 1 millimeter, the center gets out. So yes, it is necessary to make those artificial channels to get oxygen into the center of the tissue. And if we move to 3% oxygen environment, it's even becoming more important because we don't have a lot of gradient anymore. There's one question over there.
Firstly, I would like to thank you for your for your speech. It must take a lot of courage to do such a study about something so not ever studied before. Thing. I was wondering if you somehow find a way to use blood vessels or some other way to feed the muscle. Are you planning to use tendons to pull the muscle fibers, or are you going to stick with the silk fibers?
Right. Interesting question. I don't think we particularly like tendons in our meat because it tends to make it very tough and they're not very digestible. So if we can stay away from tendons, I would certainly do that. So far, with the attachment systems that we have used, which are basically sort of replacements of tendons, if you like, that has not been necessary. So they do pull themselves apart from the tissue, but from the ankle points, but it's in a relatively late stage. So I don't think it's necessary. And what I think is more important eventually, but that's something where we're doing some research on, is that these ankle points need to be flexible. Right now they're completely stiff, but they need to give way in an elastic way to account for oscillatory tension development in the muscle cell, which might improve the whole system even further. But that would be the way I would go. I would not try and make a tendon.
Because I'll ask that because if you somehow manage to do a 3D muscle, a very large muscle, it would be a little bit hard to extract the silk fibers from it also.
Right. And currently we are no longer using the silk fibers. Actually, we are growing them in donuts so that they attach to themselves. And we can grow them, I didn't show it, we can grow them on rods, a number of donuts on a rod with a thermo-elastic polymer, and so we can harvest them pretty easily from that rod. And that also creates the elastic attachment that I was talking about. Referring to the courage, I'm not a very courageous person, but I think everybody in research who wants to do something new inherently has to be courageous. There's no question about it. If you're not at least the slightest courageous, then get into something else, because this is what you need. Right.
Thank you very much.
Just one technical point that is related to the fact that you need large-scale cultures. Okay? And we all know that culturing a huge number of cells requires large dishes and everything, and manipulating huge amount of medium, and that there is always a risk of contamination. That is, and we all do, using antibiotics to prevent bacteria from contaminating the media. Therefore, there will be traces of antibiotics in the meat, systematically in the meat that you're going to end up with.
Right.
Is it going to be a problem?
For now it is. Are there solutions? I think so. Right now in tissue engineering, the first robots have been engineered so that you basically put your biopsy in one end of the robot, and it is one self-contained culture system. It's about this size, all in glass, and it's used for skin right now. It's developed in Germany. And you basically put a biopsy on one end, and the tissue engineered skin comes out at the other end. And it has all the processes of quality control, extraction of cells, proliferation, seeding on gels, the whole thing contained in a glass structure. That doesn't really completely prevent contamination, but it reduces it tremendously. The other thing is, and we haven't done an experiment yet, but I have some experience with bactericidal peptides from the catalycidin family. And we are going to experiment with a number of those catalycidins to see if we can replace the antibiotics with pretty innocuous, about 11 to 30 amino acid peptides that are naturally antibiotic. But it's in an infancy state.
Thank you for your presentation. Now we will present your certificate of true service granted in your name in the 7 trees forest.
Thank you. Thank you very much.